is transposase-based genome editing, advantage and delivery to treat retinal genetic disease. The FIP envisions a world where everyone has access to safe, effective, quality, and affordable medicine and health technologies. So our mission is to support global health by advancing pharmaceutical practices, sciences, and education. So we are pleased to deliver this webinar as part of our ongoing efforts to share relevant, scientific, and professional insight with the global pharmacy community. So, yeah, my name is Aini, and I am honored to be your moderator today. I currently serve as the FIP Early Career Pharmacy Group, or ECPG, as Industrial Pharmacy Section Liaison, and I'm also joined by Dr. Mathew Cherian, Vice President of Operation and R&D at Revive Biotechnology USA, who will be co-moderating this session. We are truly excited for the discussion we are about to have and look forward to your engagement throughout the webinar. Next. So some announcements that we have. Now we have two speakers, Dr. Sandip Nima, PhD, and also Dr. Subro Guhat Hakurta, PhD. Before into that, there is some announcement that the webinar is being recorded and live streamed via YouTube. And also the recording will be available on our website, www.eventsfip.org. You may ask questions using the question box provided. And also you are welcome to provide feedback through webinar@fip.org. And also, if you haven't become a member, there is some beneficial, become a member of IP. And then also, there is some new translation feature on Zoom. The step 1, you can see on the below screen, turn on caption. And then step 2, you can enable the translation toggle. And then the step 3, you can choose your preferred language under my caption language. And then you can follow along with the live translated captions. If you have any problem, you can just drop on the chat if you have some kind of trouble to activate this translation feature. So the today's program is introduction, and then speaker, and then presentation, Q&A, and closing remarks. Learning objective of today's webinar is understand how transposase-based genome editing technology works and its advantages in gene therapy, how lipid nanoparticles for DNA and mRNA co-encapsulated are developed and optimized, and also appreciate and comprehend in the in vivo tolerability and expression in photoreceptor to demonstrate successful delivery. And I want to introduce our speaker. First is President Biopharm CMC at Pfizer USA, Dr. Sandip Nema. Dr. Sandip Nema is President of Biopharm CMC at Pfizer and bring over more than 30 years of experience in pharmaceutical development. He has held senior leadership roles at Pfizer, Tessera Therapeutics, and Cellular Gene Therapeutics, where he contributed to advancing gene therapy programs and developing drug products across multiple modalities, including monoclonal antibodies, mRNA, and vaccines. His work covers formulation, delivery technologies, and CMC strategies, which are essential for enabling innovative therapies. And our second speaker is Dr. Subro Guhat Hakurta. He is consultant from USA. He is a biotechnology executive and scientist with over 15 years of experience advancing genetic medicine platforms and therapeutic programs from discovery through IND readiness. He has led cross-functional teams developing next-generation genome editing technologies including engineered transposase, CRISPR, and also LNP-based delivery system for neurological and ocular disease. His work has contributed to patents, publication, and collaborations with leading institutions worldwide. So, we are truly honored to have them with us today to share his expertise. So please welcome Dr. Sandip Nema and Dr. Subro. So the floor is yours. Thank you so much.
Hello, everyone. Welcome. Today, I'm going to first introduce about the genome editing. And before going into the transposase-based genome editing, I would like to introduce the concept of genome editing and what is transposase transposition and how we are harnessing transposase transposition for the human gene therapy. So, as we all know, genome editing is a technology that enables researcher to modify genomic DNA of a particular choice and replace the faulty sequence with the correct sequence very specifically. It's a sort of a molecular scissors that can, cut and remove or replace the faulty code and then replace that with the correct code in the genome. So initially, these are the three pillars that you can see in the screen that were considered as a pillar of the genome editing is the CRISPR-Cas9, TALEN, and zinc finger nucleases. In all these three technologies, what is common, it's like a bipartite system or the two-part system. One that recognizes the DNA that need to be edited, which is mediated by, in case of CRISPR, a guide RNA. So it's a RNA DNA hybrid that is formed. And then in case of TALEN and the zinc finger nucleases, it is a protein that is designed or programmed, a stretch of amino acid that recognizes the bases. Now, the second part of this genome editing tools is the nuclease. In case of that makes the cut for the remove, replace everything. So in case of CRISPR-Cas9, it is the Cas9, that nuclease that works for cutting. For TALEN, it is the FOC1 nuclease. And for the zinc finger nuclease technologies, it is the ZFN nuclease that works in that. So in all these technologies, what is common is that it can cut and replace a single base pair and replace it with the correct base one. So based on this, I think their world has moved, and then we developed several different newer technologies for the genome editing, which I will briefly introduce. I made a table for you all to skim it through. So these are like base editing, prime editing, CRISPR Cas12, Cas13 system, and the transposase-based genome editing, which is the topic of our discussion in detail today. So for the base editing, it is nothing but an editing system where a CRISPR nickase, Cas9 nickase is used. What is nickase? It is mutated in such a way that it can cut only single strand of the DNA. So Cas9 nickase is fused with the deaminase that can chemically modify single base pair or single bases. So cytosine to thymidine, adenosine to guanosine, like that, and correct the base. And the next one is a prime editing, the most complex genome editing technique and very evolved. In case of prime editing, you also use a Cas9 nickase, and that is fused with a reverse transcriptase. And they also have a very specific guide RNA called prime editing guide RNA or the PEG RNA. PEG RNA contains the spacer sequence that search for the sequence where need to be edited. Then there is a primer binding site, and then there is a reverse transcriptase template which contains the edited sequence which will be copied into the genome. In this technology, it's the most precise technology, and but it requires many more systems to be involved to make the editing. On the contrary, CRISPR Cas12, Cas13 system is a very simple system. Cas12 has the evolved PAM sequence. It doesn't depend on the NGG PAM sequence. Whereas the Cas13 system is even beautiful that it can directly edit the RNA instead of the DNA. No DNA is required for the editing. And then lastly, the transposase based genome editing, where you can insert a large piece of genomic DNA or a gene size insert into a specified sequence, or a tetranucleotide sequence. For suppose piggyback, it is TTAA. For sleeping beauty transposase, it can go at a TA site, and you can introduce a whole large sequence in the genome. Many clinical trials have initiated for this, for base editing. You all know that Verve Therapeutics is now acquired by Eli Lilly, is doing the PCSK9 gene editing, a single base pair gene editing, and lowering the LDL cholesterol for familial hypercholesterolemia. Beam Therapeutics, which also originated from Dr. David Liu's lab at MIT, they are also doing sickle cell disease and the beta thalassemia editing. For prime editing, the Prime Medicine is doing CGD, chronic granulomatous disease, by editing the NSF1 gene in the CD34 positive hematopoietic stem cells. In case of CRISPR-Cas12, the Editas Medicine is doing using CRISPR-12A, Vital gene is using CRISPR-12B, and Hubida gene is using CRISPR-13. Poseida Therapeutics, on the other hand, using a piggyback-based CAR T therapy for metastatic castration-resistant prostate cancer and the CAR T therapy. Now, what is transposition and transposase? Very briefly, transposition is the moving of a gene segment from one location to another location. This is called transposable element, and the enzyme that helps in moving that section of the gene from one location to another location is called the transposase. There are two types of transposition event. One is class 1 transposition, another one is class 2 transposition. Class 1 transposition is the retrotransposition, which is copying the segment in the form of RNA, then the reverse transcriptase forms a DNA intermediate, and then gets copied into another section of the genome. Whereas a DNA transposition, or the class 2 transposition, is a cut and paste transposition, where the transposase recognizes the DNA element or the transposon, which is flanked by the TIR, which you can see here, this green bar, and then they excise it out and then paste it at a sequence that is guided by a tetranucleotide sequence like TTAA, or a dinucleotide sequence like TA elsewhere in the genome. And usually a transposition event is marked by the target site duplication. So where it gets integrated, that TTA is replicated or duplicated on the other side of the transposon. If you do a genome sequencing, you can easily understand whether the transposition has happened. A sequence need to be flanked by the TTA on the both sides. So based on that, as I told you that there are two different types of transposition happens. Transposase are differentiated based on their catalytic domain. In case of class 1 transposon, it is only reverse transcriptase integrase. The one famous example is the line 1 L1, which is being right now harnessed for the gene therapy. And for the class 2 transposon, there are DDE transposases, which I will be talking about today, like piggyback transposase, sleeping beauty, piggyback, HUH transposase, and serine and tyrosine transposases. So how we are harnessing transposase for transposition method for our gene therapy. So as I told you that usually the gene that section of the gene that gets transposed to somewhere else is marked by two ITR or TIR sequences, or the terminal inverted repeat sequences at the end. What scientists had done, they just took that kind of a TIR sequences and flanked it by the cargo or the gene of interest that you want to insert in the genome. So your gene of interest is now flanked by the two TIR. When you co-overexpress that in the cell with a transposase, that transposase excises it out from the cargo from the donor DNA plasmid, and then it integrates at the TTA site where it can be integrated. This kind of integration could be targeted integration as user specified TTA, or it could be a semi-randomly any TTA that it can be integrated. Both type of gene therapy has values which I will be discussing. Now, at a cellular level, what happens, what we had done at previous company Salugen, we never wanted that transposase could get integrated into the genome during the gene therapy. Because if it does, then it will constantly remobilize the section like a jumping gene. So we wanted the limited exposure of the transposase. So we coded the transposase or in the BMLT, or the bioengineered myotis luciferase transposase that we are working with, we coded that in the mRNA form. And then the donor DNA is in the plasmid. We encapsulated both of them in the lipid nanoparticle. When upon entrance, the transposase gets translated immediately because it's a mRNA, right? Translated into the cytoplasm, and then it has an intrinsic nuclear localization signal. It enters immediately into the nucleus. On the other hand, the donor DNA plasmid also augmented with the NLS sequence. They go into the nucleus independently, where the transposase binds to the transposon and excises it out and integrates at the TTAA site. Now, you probably notice that I always draw the picture of the transposase, the BMLT, as a dimer. So we confirm that by the cryo-EM structure. Here, if you notice, you can see that the two monomers are interacting in a cell-free system always. And when we develop the sequence, it will almost look like this. It is modeled after the piggyback, but this is the interference amino acid that are binding the two monomers together. Now, little bit of a data. We wanted to see whether as I told you, that transposase protein, you do not want it to be very long standing because otherwise it will be constantly remobilizing it. We wanted to check whether the mRNA in the cell is present for how long, and we confirm that they are disappeared by 72 hours, so as the protein. And we also measured the integration activity using our specific assay that can measure excision and integration. We also modified the MLT transposase in a proprietary way so that it can be very hyperactive. And we saw that it has very high integration activity at the TTAA site. Now, I told you that they have high integration activity. So we wanted to see whether they are very good at integrating at the TTA sites and where those TTA sites are that are being targeted by the BMLT. And very surprisingly, or very fortunately, what we found that the majority of the integrations are happening at the intronic TTA sites or the intergenic TTA sites. Very little percentage of those integrations are going either exon or any meaningful sequences. Although, so that definitely calls for a very good safety feature for this gene therapy technique. But then, because some of those events are happening still in the exon that can disrupt the gene, we wanted to see that how many such integrations are happening at the one particular locus at the TTA site in the exon or meaningful sequences. We found that majority of those integrations are happening only one time. So knocking out two alleles of the gene are not happening randomly. So the safety feature is very high, and we went with to the regulatory filing agencies, and they were very happy with this profile. Now, I want to say that why this kind of gene therapy would be important. So one of these gene is ABCA4 gene, which is a very, very large gene. And this gene actually has almost 1200, more than 1200 mutations reported. And that could be linked to the disease. What this gene does, the protein of this gene actually clears the toxic product that gets accumulated in the photoreceptor cells during the visual cycle. If those do not get if the gene is mutated or partially functional or dysfunctional, then they will not be removed, and the retina photoreceptor cells will start to degenerate. And that is what a Stargardt disease is. You can see the retina, the normal healthy retina, and the disease retina. Lot of integrations are happening. Now, as I told you that these are many mutations are responsible. So if you employ base correction by CRISPR-Cas9, then you need to employ many different, CRISPR-Cas9 machinery to correct this. Instead, if you use the transposase-based genome editing, you can just completely recruit the functional copy of this gene into a TTA site, and because it gets integrated at multiple sites safely, you can get a very higher expression and reverse the pathology. Now, I will also briefly want to introduce the targeted integration, how it can happen, because it can integrate at many TTA site. The main challenge for the targeted integration using transposase is to how to reduce it any other integration apart from a TTA that is designed by the user. So we did that. We fused a DNA binders on the each side of the dimeric transposase that are programmed to target the specific TTA, and also mutated the intrinsic part of the catalytic domain of the enzyme, so that it becomes completely dependent on the target recognition on the DNA binder. So now you have the donor element that you want to introduce, one transposase on the left side fused with the DNA binder, and one on the right side of the target TTA. All those three complex can be put together into the cell, and they will bring the cargo at the particular TTA site that we want to introduce. By doing that, we can go either in a genomic safe harbor site, or we can go to the native locus where the faulty gene is. But it will have very low expression as the gene should have if they were functional. Okay, so this is how we achieve the targeted integration. We could not go any further for that because of the funding crisis in the company, but we achieved quite a lot using the transposes both at the targeted TTA as well as integration at any TTA. So with doing that, we actually reduced almost 30-fold non-targeted integration of the cargo at any other genome, any other place in the genome, but we increased almost 200-fold targeted integration at the particular TTA site, and we compared that when we compare simultaneously transfected the untargeted BMLT and the targeted BMLT. And we found that the targeted BMLT could introduce almost 200-fold more than the untargeted one. So, in the summary, I want to say that the transposase genome editing enables stable and permanent insertion of a very large fragment. And because this BMLT is from the little brown bat or Myotis lucifagos, it's a mammalian origin, as well as it doesn't cross any double strand break at the site of integration or during the transposition. It is very safe and immune reaction is low. And by doing the mRNA based delivery, we also ensure that there is no genomic instability because transposes do not get integrated into the genome. And the targeted mediated integration also allows the functional gene copy at the native locus under the same epigenetic control. So there are a lot of advantages of the transposase gene based genome editing. And now I will hand it over to Dr. Sandeep Nima, who will cover the delivery section of this beautiful gene therapy. Thank you.
Thank you, Subru, for a very nice overview of the transposase-based gene editing and what advantages it has. So just briefly, what Subru stated, and I wanted to provide a little bit more color to that, is that Stargardt, the gene which becomes defective in a Stargardt is a big gene. There could be thousand mutations in there. So if you are using a CRISPR or base editing, you may need 500 or 600 or several hundred at least different drugs to treat all the patients with different mutation. But with this technology, you replace the whole gene, so one therapy can treat all the Stargardt patients. So that's one of the significant advantage of this technology is. So when we were looking at how to deliver the transposase-based gene delivery, we were looking at there were two possibilities. We could either use an viral vector to deliver these or a non-viral system. We went with the non-viral system for several reasons. First, as I mentioned, the size of the gene to treat Stargardt is big. It would not fit an AAV viral vector. Therefore, that was not also a possibility. Also, the non-viral vector have advantages over the viral vector system in being non-immunogenic. You can also do repeat dosing. They are more versatile. There is better manufacturability, and therefore the cost of goods for making them is much lower than a viral vector. So we decided to go and look at the non-viral delivery system. We selected lipid nanoparticle as our lead technology to deliver the transposase-based gene therapy. So the way we approached the LNP formulation screening to deliver the drug to retina, we established first our goals. What were our goals? One was to make sure that the LNP system can deliver mRNA, which encodes for the transposase, and DNA, which is the gene for the Stargardt disease. So both of these have to be co-encapsulated into the LNP. As Subru showed earlier, once LNP is taken up by the cell, it goes into the endosome. So you want to make sure that the cargo, which is mRNA and DNA, both are released from the endosome. And also DNA needs to move into the nucleus where it can get integrated. So we want to make sure that is happening. Obviously, stability is important for both the nucleic acid and also since this drug is going to be delivered into the retina, we want to make sure all the excipients are going to be compatible with the subretinal application. So the strategy for developing the mRNA, we first use some surrogate nucleic acid. In this case, we use a GFP mRNA and GFP DNA. So GFP stands for green fluorescent protein. And the beauty of that system is that you can easily visualize it. It's a good system to screen it. And once you find out which LNPs are better, then you can actually put your real cargo in to look at the immune responses, to really look at the in vitro and in vivo studies that you can really do. So the experimental part was twofold. We did in vitro study, screening studies using a cell line, which is ARP19, that's the retinal pigmental cell epithelial cell line. You are looking at tolerability by looking at the viability of the cell and also expression. Then the best formulation were screened in vivo, and this was done in mice, either normal mice or when we were looking at the actual disease treatment, we used a transgenic mice which had ABCA4 gene knocked off. So it was a knockout mouse model, and we wanted to see now in this model if the ABCA4 gene is going to be expressed or not. So this is what I'll describe briefly what we did. First is we manufactured at a research scale, at a small scale, but we wanted to make sure the process can be scaled up. In fact, we were able to successfully scale up this process, but I will not go and talk about it. The LNP manufacturing process started with the lipid mixtures. In this case, ionizable lipid, the helper lipid, the PEG lipid, cholesterol, and others were dissolved in ethanol. And the nucleic acid, both mRNA and DNA, since they are very charged, were dissolved in low pH buffer. The two streams were mixed using a microfluidics. There are a bunch of different equipment available. We use nano assembler. And as these two streams are mixed together, the polarity of the solvent, in this case ethanol, changes and the lipid precipitates into the form of lipid nanoparticle, co-encapsulating both mRNA and DNA. So the actual event is really the ionizable lipid at low pH is positively charged, and the nucleic acid, as we know, are negatively charged. So they form a ionic complex which precipitates out, and then you do diafiltration to remove the ethanol, and any unencapsulated nucleic acid. You do buffer exchange into the by for concentrating, changing it into the final formulation. You filter it through zero point 0.2 micron filter and put it into the container. And this is what is used for either doing in vitro or in vivo experiments. So this is a cartoon of um, the lipid nanoparticle in which you have the mRNA, which is coding for transposase, which is BMLT. You have the mini plasmid, which is the gene for Stargardt, and the other lipids, which makes the LNP. So as I said, the first step was to screen these LNPs for using in vitro cell line, which is the ARP19 cell line. As I mentioned, it's a differentiated cell line. It's a normal cell line, so it's not a disease cell line. What we were looking is use a 96 well plate experiment, and we on the cells we put in the LNP and look for the cell viability, which is shown on the left hand side of the graph. So after at different doses, see if it is tolerable or not. And the second, we also look for the expression. In this case, we are looking for green fluorescence protein being expressed. As you can see, here we use a variety of different ionizable lipid. You can see some of these lipid nanoparticles were much more tolerated than others. So when I say tolerability, that means the cell viability was high. And then you also see differences in the expression of the amount of GFP. So those which were expressed more and which were tolerable were the one which we selected. Now, this is just only a few of the ionizable lipid. We looked at a bunch of um, others also, but this is just a representation of some of them, what we did. Then we went and looked at the in vivo studies. So several of the best in vitro formulation were looked for in vivo study. But before I describe how we did the in vivo study, let me take a step back and show you the structure of human retina. As you know, the human retina has different layers. The basal layer is the retinal pigmentosal epithelial. Then you have an outer nuclear layer in which the photoreceptors, the rods, and the cones are present. So these are rod and cone in the outer nuclear layer. And then you have an inner nuclear layer, and all these nerve fibers then join together to form the optic nerve, which goes into the brain. So this is the structure. We are interested to make sure that the gene is expressed in the photoreceptor, not so much in the retinal pigmentosal epithelial, because this is where it needs to function. So the way we look at the retina in vivo is we inject 1 microliter of our formulation into the subretinal space of the eye. We looked at the, after the mice have been injected over a matter of days, we look at the OCT scanning of the retina. Basically, we are looking at the thickness of outer nuclear layer, whether it maintained it or not. If they are cell death, then this layer thickness will decrease significantly. If these are intact and healthy, it will maintain it like a vehicle, if it was injected with a vehicle. So that is in the live mice looking at the tolerability. The second thing we also look at is the expression of the protein by looking at the fundus. So this is just, is the green fluorescence present or not, and some sort of a qualitative estimation. So we can quickly determine which of these formulations are still doing good. So our goal is do a lot of in vitro screening, a few in vivo screening, and then really then start selecting and optimizing it. Now, when I tell you that in the fundus imaging, you see that there is a green fluorescence and in the retina, but what you really don't know is where that green fluorescence is present, whether it is in the retinal pigmentosal epithelial or the rural photoreceptor. So you take the retina out, you dissect it, and then see where that green fluorescence is by quantitating the green fluorescence protein. You want to make sure the dissection is good, because if it is by looking at rhodopsin as a biomarker, here we looked at the Western blot to confirm it, because if it is in photoreceptor, you see a lot of rhodopsin, but in ICUP or the RPE, you don't see it. So we wanted to make sure that the dissection is good and quantitatively measure it. So we did this formulation screening, the analytical characterization, and other studies multiple times to select the best formulation, um, a best ionizable lipid. Then we also try to optimize the composition, because as you know, ionizable lipid is not the only component. You can have different helper lipids, you can have different cholesterol, different PEG lipids, you can have different composition. We looked at all of that and used the same sort of a screening technology that I described earlier of in vitro and in vivo in stepwise fashion to select the optimized formulation. In the end, the optimized formulation had not only good tolerability, but it improved the expression several folds compared to the non-optimized LNP formulation. We also looked at a targeting agent, because we thought that if I can attach a targeting agent like vitamin A, it may have a better way of going into the photoreceptor. So we looked at that study, but it did not provide any additional benefit. So we did not use it in our final formulation nomination. So this is what the final LNP looked like. It had the transposase, which is BMLT in the form of an mRNA. It had a plasmid DNA, which encoded for Stargardt gene in the form of a DNA. You had ionizable lipid, phospholipid, cholesterol, and PEG. And this is the LNP. The size was around 80 to 90 nanometer. So another thing that we need to really look at is to characterize these LNP and have a good idea of their properties. The way we ended up doing is, besides using the traditional common technique of dynamic light scattering and measuring the size, the polydispersity index, the zeta potential, we also, and the encapsulation efficiency using ribogreen, we also looked at the payload distribution, the surface properties and morphology of the particle distribution by variety of techniques. And I'm going to just select one because we have limited time. So we used AF4. This is the asymmetric field flow fractionation coupled with mass to look at the size even more carefully, because it is better able to have separate than dynamic light scattering, which is more of a bulk property. We looked at the structure and morphology of LNP using cryo-TEM and SACS. And we looked at the payload distribution. So the payload is mRNA and DNA into the nanoparticles. So how did we do that? Because so nano FCM was the technique we use, which is it's a flow cytometry technique where you look at not only scattering out the light, but you can label these lipid nanoparticles with fluorescence, and then you look at the fluorescence in different channels. So this can go down to size of about 30 nanometer, and it looks at individual particles, not the bulk particles. So we labeled the nucleic acid with cyto 9 fluorescent dye and tried to see what do we see? So if you have just empty lipid nanoparticles, you don't, it says 99% empty. We obviously optimized and qualified this technique. And then our product, which had both DNA and mRNA, what we were able to confirm that 98 to 99% of these particles have nucleic acid present in them. So we only have about 1 to 2% or less than 1 to 2% of the particles which are empty. And that's important because you want to make sure that you have most of these particles, if they are taken up, can deliver nucleic acid. Finally, after characterizing and selecting the best lipid nanoparticle, we did the experiment in an ABCA4 knockout mouse model. We injected 1 microliter of these optimized LNP in the final formulation. And then this was done at 2 different doses to see the impact of those. So couple of things that are apparent here is that OCT, which is the ocular coherence tomography, show good that the product was tolerated well. We also see that approximately 35% of the photoreceptor express ABCA4. So that's very good that you actually see the expression in the photoreceptor. And on the next slide, I'll show you why we know that it was expressing in the photoreceptor, because you can look at the entire retina and get the numbers, but you don't know which cell type, particular cell type it is expressing it. What I have wanted to point out here is based on our calculation, we needed only 15% of the photoreceptor to be expressing ABCA4, and this amount would be sufficient for correcting the disease in the human. So that was a very good, and that's a very good data which shows that this technology has extreme promise. Obviously, the mouse data may or may not always correlate with the human data, but very promising data. So this is immunohistochemistry. So we did immunohistochemistry of the retina to show where the ABCA protein is expressing. So let me orient you to this slide. The purple, which is the bottom one, I'm not sure if you can see my pointer or not, is your retinal pigmentosal epithelial. The next layer is your outer segment with the photoreceptor. So we used a antibody against ABCA4 protein, and it shows green color, and the green color is in the outer segment where the photoreceptors are. So we knew that the ABCA4 gene is expressing predominantly in the photoreceptor, almost none in the retinal pigmentosal epithelial, and the blue color is the DAPI, which just stains all the nucleus in the cell. So this data shows that precise restoration of ABCA4 protein occurs in its natural photoreceptor outer segment location. So in summary, the transposase-based gene delivery offers several advantages over CRISPR or base editing, including no double-stranded break. You can replace the entire gene, so all the mutations can be corrected, not some or some of them. What we were able to show today was that we had a in vitro and in vivo screening technology which was used to optimize the LNP system. The LNP encapsulated the transposase, which was in the form of a mRNA, and a large DNA coding for the full-length human ABCA4 gene. This formulation exhibited excellent pharmaceutical characteristic in terms of being well tolerated in the mice after subretinal injection. And after treating with LNP, which encapsulating the transposase of BMLT mRNA and ABCA4 DNA, the mice showed significant protein expression. And with the IHC data, we showed precisely that the ABCA protein expressed in its natural photoreceptor location. So the results show the feasibility and tolerability of subretinal administration of a non-viral integrative gene therapy to deliver a large gene to photoreceptor in Stargardt mouse model. And with that, I'll conclude and thank you very much for attending and listening to the presentation.
Thank you so much for the insightful presentation from Dr. Sandeep Nema and Dr. Subro. Now we have several questions, and I want to read it. The first question is from Luis. Maybe for audience who may be new to this field, like maybe could you explain how does genome editing compare to traditional gene therapy for retinal disease? Maybe for
Sure.
Dr. Subro.
Sure. That's a very good question. Um, so the traditional therapies or the traditional gene therapies are mainly started with the AAV based or lentiviral particle based gene therapies, which are non-integrating technologies. But in case of the genome editing based technologies that are for retinal therapies or something, first of all, if you are editing a base in the DNA that is permanent change, and there will be no other extra machineries will be left in the tissue or in the cell. So that is a big win. As well as I think the second point is also a little bit correlated. This is a one shot therapy, right? So once you make the change, it will stay forever, and the patient has to undergo this kind of therapy only once, and they can have the benefit for the rest of their lives. I think that is the biggest. And genome editing based therapies, if it is targeted genome editing based therapy, it is very precise, and it doesn't touch anywhere else in the genome. And if it is untargeted one, it is also like as Dr. Sandeep Nima presented, and some of my genome wide sequencing data showed, even if it is untargeted, it usually integrate at a specific sequence of TTAA or TA, but also they are in the non-interfering sections in the genome. So they are not usually hamper the gene functions. So this is definitely a difference compared to the other therapies.
Thank you for the answer, doctor. And then we have another question. What specific retinal disease are most likely to benefit from this transposase-based genome editing?
Right. So I can take the answer, and then if Dr. Nima feels he can add more. So I
Yes.
think one example we already presented together is the Stargardt disease. Any gene that are not any disease, retinal disease or any other disease per se, that are not caused by only single mutation, I think can greatly benefit from this kind of therapy. Whenever you have a gene which has many mutations are there, which could be linked to the disease, you can apply this kind of gene replacement or gene augmentation therapy by transposase.
So the only other thing I wanted to add is the same technology we were applying for other diseases. We just presented today the retinal disease. So this technology or transpose-based gene editing can be used for any genetic disease where you can edit the gene or replace the gene. And I think the bigger challenge is also if you can deliver it to the right tissue or organ, that becomes the case. But in theory, this should apply to any genetic disease.
Okay, thank you so much for the great answer. And then there is question like, hello, may I know if this article has been published? If yes, kindly provide the publication link.
Yeah, we have not published it, but we have presented it at conferences, including I know ASGCT, Control Release Society, and other places, but not
Arvo.
really. And ARVO,
Arvo. Yeah.
which is the Ophthalmic Society meeting. So those are the places where we have presented but not published it.
Okay, thank you so much. And then next question for Dr. Sandip Nema. What are regulatory challenges exist in approving genome integrating tools for retinal disease compared to episomal gene therapy AAV based?
Yeah, so the biggest thing is not so much from the CMC perspective, but more so from the safety perspective, because here you are permanently integrating the gene into the genome. So you want to make sure that as you integrate it, there are no that it is being integrated safely. There are no double-stranded break. If they are double-stranded break, what could be consequences of those? So really, the safety becomes the major thing because it's going to be irreversible, permanent, and so that's why FDA has been much more cautious and trying to do it more on a risk-based analysis. So diseases which are more severe, there is more appetite to allow that to happen. But I do think the safety is the biggest thing. The second thing is you know, AV has its own challenges because if you have a pre-neutralizing body antibodies to some of these AAV-based gene therapy, the patient cannot get it. And for any AAV serotype, approximately 20 to 40% of the patients may already have a pre-existing antibody. So you cannot give it to that therapy. So that's why the non-viral delivery system have significant advantage because that's not a concern. You can repeat dose it. So let's say if you see you want a 50% correction and the first time you only get 30%, you can do it one or two more times by LNP because it's a procedure that can be repeated either IV or in tissues, and then you get the expression where that's not yet possible with AV. So I think the safety related things are the biggest challenges. Um, AV also you have to do viral shedding studies to look at the safety, and these type of studies are not needed for a non-viral based gene delivery.
Okay, thank you. So, yeah. If you have any additional, Dr. Subro?
No, I think I'm good.
Okay. 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 Next, the question maybe can deliver to both of you. Like, can genome editing be used for all types of inherited retinal disease? Yeah.
Yeah, so I think I can answer it first. I think all type is a very strong word. So I think majority of the genome editing based therapies, like genome editing is a very big word, right? It encompasses the base pair change type of genome editing versus a small deletion or small insertion correction versus a full gene which encompasses many mutations or substitution unwanted that you replace or you augment that a functional copy. Having said that, I think uh, majority of the diseases that are being currently looked at by gene therapy can be addressed by genome editing, if not all. I cannot really say that all the gene retinal diseases could be answered by genome editing, but majority of the diseases which caused by the gene mutation, which are caused by the substitution, deletion, or multiple mutations at different gene, all of them together. Suppose in case of I can give you an example. Prime editing could be a method of choice where multiple genes are need to be corrected at the same time. So yes, majority of those could be answered by genome editing, and the large substitution could be answered by, I think, transposase based editing as well. Thank you.
I think, Subru, you have pretty much answered what I would have said anyway, so I don't have much to add to this.
Okay.
Okay, thank you so much. So, we don't have much time left. So maybe this is like we are talking more about the future. This is like a forward-looking question. First from Dr. Guhat Hakurta. Beyond Stargardt disease, what other genetic conditions are you most excited about tackling with this platform? And maybe Dr. Dema, how adaptable is this LNP system? If you wanted to target a different organ like the liver or the central nervous system, how much of the formulation would have to be reinvented from scratch? Maybe the last question.
Okay,
Thank
so I can go first. Thanks, Aini. This is a very interesting question. Yes, I think apart from, as Dr. Nima previously said, I think we also try different type of disorders that we can tackle with this kind of a transposase-based editing. One of the diseases that we wanted to really find a solution for was a cystic fibrosis, by targeted integration of the whole cystic fibrosis gene or the CFTR gene at the endogenous locus. And we were achieving much success at that, but prematurely terminated by that in the company. However, I think apart from those, I think as I mentioned, any diseases, any genetic or inherited diseases that encompasses multiple mutations, or if you need a very good expression of the corrected gene, those kind of diseases could be answered using the transposase based therapies. Absolutely. So one example is a cystic fibrosis, which also has over 2,500 mutations in in that particular gene. Then another one, I think I could say any sort of a liver disorders that are, I think, are being currently checked, could be answered by that. And also the exogenous ex vivo therapies, lot of CAR T therapies also could be done using transposase based editing. Thank you. Sandeep, you want to add anything to that?
No, I'll just answer my part of the question
Okay.
because I know we are running out of time. Is so the fundamental principles of the LNP characterization and manufacturing apply, but the formulation, the ionizable lipid, the composition, and sometimes you need to have a targeting agent if you are going to target it to a specific cell type has to be incorporated. So I would say that some of the principles do definitely of LNP applies, but then you have to tailor make it to different cell and organ based on what the need is. Now, hepatic targeting has been achieved, and it's probably easier, but targeting it to specific hematopoietic cells or other cell types is still being developed, and you sometimes have to attach an antibody or a fragment of the antibody or other peptides to target it to the cell so that the LNP can go over there. So it is a not as straightforward as taking the exact same LNP and applying it or just making some minor changes. Either it could be if it is a closely related tissue, but it would require significant amount of formulation development work if you are looking at a completely different cell type.
Okay, thank you so much. Thank you so much, Dr. Sandip Niyama and Dr. Subro, for sharing your expertise in this very inspiring ideas and research area. And thank you so much for everyone who attending the webinar. If you have any feedback about today's session, if you have any unanswered question, you can send email to webinars@fip.org. So thank you so much.